Okay, welcome everyone once again. My name is Adrian Vasquez, and I'll be moderating today's webinar. Um, welcome to our Water at Wayne webinar titled Integrative Ecology of Green Architecture and Blue-Green Infrastructure for the Recovery of Ecosystem Services in Urban Environments. Before we get started, I just wanted to go over some housekeeping items for the webinar. Throughout the webinar, you will be muted as participants, and please keep muted. You'll also, if you would keep your videos off so that we can just focus on the speaker. And you can ask all your questions in the chat box throughout the event, which we'll deal with at the end. So it is my privilege today to introduce my colleague and friend, Dr. Victor Carmona. Dr. Carmona is an associate professor in the biology department at the University of Detroit Mercy. He is the director of sustainability at UDM, the College of Engineering and Science, as well as a research associate with the Center for Eco-Health Research, CEN Salud, at the University of El Salvador in Central America. That is one of the first places that I got to spend some time with Dr. Carmona, so I know him, working with him with his connections in El Salvador. Dr. Carmona has also served as a Fulbright Scholar and Fulbright Flex Scholar. Presently, he serves as a Fulbright Specialist. He has collaborative undergraduate research programs in Costa Rica, Nicaragua, El Salvador, Mexico, as well as the US. And Dr. Carmona's research students have successfully launched international collaborations via the Fulbright Student Program and Engineers Without Borders. I personally know Dr. Carmona and work with him in many projects here in Metro Detroit. And we have many more plans. So I'm really looking forward to his talk. And I hope you are too. So let's pass it on to Dr. Carmona. I think you're muted, Dr. Carmona.
Yes, I am. Sorry about that. I just thanked everyone. I'll thank you guys again. Adrian, thank you so much for the warm introduction. And also, I wanted to thank everyone at Wayne State for the invitation for today. This was a talk I've been wanting to put together for a while now as Director of Sustainability at the University of Detroit Mercy. So this was the perfect venue to kind of showcase some of the ideas that I've been working on. What I want to do today is kind of start with some introductory topics here where I'm going to kind of change a little bit the way that we are defining sustainability so that I can add this layer of integrative ecology, and we'll make some sense of what that means. I will borrow a lesson here from climate change because I think it's important to think about species in terms of how they interact with the environment rather than as an entity of itself. And of course, to do a proper introduction, there's some terminology that I'm going to introduce just so that we all are on the same page. But really what I want to do during the talk today is highlight some of the regions, areas, spaces that I work in. These are going to be different topics relating to forests, urban water, as well as urban agriculture. And they're going to span all over. They're going to be in California. They're going to be here in Detroit. They're going to be in Costa Rica and even through in some from El Salvador. But the idea is to have it topic based. So I'm going to go through a whirlwind of different projects. My hope is that you'll find a couple of intrigue and that will kind of launch into the Q&A at the end, where I'm going to share this model of collaboration, which I feel is this integrative approach that I'll be showcasing today. So the first thing I want to do is contrast this idea of operational sustainability, which is typically monodisciplinary in its nature and tends to celebrate the efficacy of tools. And I want to contrast it with environmental sustainability, which tends to be multidisciplinary and integrates collaboration. So really, as the saying goes, the problem with a tool-based approach that's monodisciplinary is that if the only tool you have is a hammer, then everything's going to look like a nail. Whereas in a multidisciplinary approach that's integrative, you're always looking at the problem with new eyes. So it never looks the same. So let's take a couple of projects here, very large projects, and kind of throw it in this integrative way. So I picked this one. This is the Kobogen building in Dusseldorf, Germany. And if you look at it, it's absolutely stunning. They just launched it opened it in 2020 in the spring, and it showcases a lot of really neat features. I want to pick out a couple of these just so that we can see how really green architecture is going to be trying to mitigate these environmental impacts that come from the building in terms of moderating the use of materials, even how much energy is consumed, or the actual footprint of the building itself, and the system, the city system with which it interacts. I'm going to make an exception here. We're going to talk about that word in a couple of slides. This is from the Kobogen website. So if we look at this feature of storing water, there's a problem here. Yes, there's vegetation and soil. So yeah, water will be stored. But remember, these are plants. So there's an evaporative transpiration that's also going to be occurring. They actually had to install a irrigation system across this building because this particular plant needs regular watering. So it's actually storing some water, yes, but it's also utilizing a lot more to maintain that green aspect. In terms of capturing CO2, true, photosynthesis is going to do that for you. But this is a deciduous plant that they went with. It's called hornbeam. It's native to Europe. And it loses its leaf at the end of the growing season. So the carbon that's captured by the plant is dropped in the leaf litter. And when that decomposes, it's going to release that carbon dioxide again through decomposition. And we also know that the cement that goes into the infrastructure is also responsible for releasing most of the CO2 that this building will contribute, much more than the carbon dioxide that's going to be bound by the plant material. And then if we look at the last one, this idea of supporting biodiversity, again, is problematic because it's a single species. This entire building is monospecific. It's a monoculture. And so that means that, yes, you've added one more species, but that's not really what biodiversity is. So we'll talk about that term as well. So what you're seeing here is that really green architecture seems to be highlighting more of this operational approach, but a beautification kind of highlight. If we contrast this with green engineering, it looks very similar. This idea of mitigating the impacts of the infrastructure with the environment. And again, here we're reducing pollution that's brought on through the construction or any hazardous materials during the construction. There's the viability issue of economic feasibility. But one component here that's mentioned that's important is matter has to cycle through it, and matter has to be recycled inside of the system, and energy has to be cycled through. So in this particular project, this is the Rio Manzanares in Madrid, Spain. And you can see that it's a riparian area that just got remade. In fact, this used to be a road. It's now a sidewalk with bike lanes. If you look at some of the highlights that it touts, again, this idea of storing water, it's the same issue we have as before. There's actually more water being used because there's actually a water feature right around the corner here. And these trees are all from Mediterranean woodland. They're going to require constant watering. And of course, the same issue we saw before with the CO2 capture. So what you start to see here, if you start peeling the layers of some of these features that are touted, is that there's a lack for a functional ecology for, in this case, a riparian system. It's just ignored. and the social justice for the local community is actually just limited to tourism. So how does this integrative approach work? Well, we're going to extend basically from an organizational level of biological diversity. And we're going to do this at several levels, but the idea is to see how those respond to global change. And when that change occurs, what impact does it have on the functions of that ecosystem? So remember, this is typical for ecological phenomena. It's going to be inherently complex because of all the spatial features that we have to deal with, going from biosphere down to the organismal level. But there's also some emerging fields that are worth mentioning because these are kind of the brave new fronts. This idea of the organisms interacting with the atmosphere. There's a lot of cool research coming out of that sphere, aeroecology. There's also, of course, urban ecology, with I think we're very familiar with here in Detroit, how organisms interact with man-made systems, anthropogenic systems. And of course, agroecology, which is the better known, where we're looking at not just the productivity in terms of the yield for crops, but also having them behave like their own ecosystem. So the way we're going to do this integrative approach is basically two steps. The first thing we want to do is study a set of species or a group of organisms and see how they're assembled to form a community. That's the first step. The second step is let's analyze the different roles that population or community plays in the functioning of that ecosystem. So what I'm going to be doing as we move forward is basically highlighting these two elements as an integrative approach. So it's going to seem a little odd, like I'm jumping you off the cliff, because the question is, now that we know the function, how do we adapt? How do we adapt it into that infrastructure? So before I jump into those examples, I wanted to highlight one clause here, kind of like a warning that something we learned as we're doing these more and more of these climate change studies and how they impact species. This one in particular has to do with a very common bird in Europe. It's a temperate bird, and it is a study that comes out in '08 that basically looks at egg laying. The timing of egg laying here on the x-axis is increasing as you move to the right. And what the study does is it goes through the literature and figures out what the 45-year average for egg laying is. And they calculate it and they figure, okay, this is where the historical egg laying date is. When they go to the Netherlands, they find that that is exactly a match for the egg laying that's occurring for this species in that part of its range. However, when they go to see the same species in Great Britain, the other extent of its range, they see that there's been a shift in the egg laying timing. So they dig up what's going on. How come one species have two responses to climate change? And it turns out that in Great Britain, the birds are emerging sooner from their eggs because the insects they feed to their chicks, these chicks that are hatching, also are emerging sooner. Europe is getting warmer, so the summers are arriving earlier, spring is shortening. So these insects emerge, the birds have to respond. But in the Netherlands, that's not the case at all. The birds are still responding to photoperiod. So here's one species with a broad enough range where the selective pressures in one end are to stay egg laying as always, and in the other are to lay eggs earlier. So we can't treat species as a one size fits all. We have to think about how they interact with that environment. That environment is going to dictate whether something is going to be benefiting that species or not. So moving into the terminology, and this is kind of hopefully a review for everyone. I just want to make sure we all use the same terms. The first term that I want to introduce is this idea of richness, because it sometimes gets confused with diversity. It literally means how many types there are, how many types of organisms are there. So if I have habitat A and habitat B, you'll notice that they have the same 5 species. So in this case, species richness is exactly the same for those two communities, but it's clear that they're not the same community. So why do we care about species richness? Well, we know that species richness can increase resilience to drought. So here's some data from biomass loss to drought in a grassland. And you can see that in terms of the number of species in the plot, there's this negative relationship. We also care about species richness because it shows resilience against invasive species. We see the same negative relationship here with the number of invading species and how many species there are in that plot to begin with. And the third reason we like species richness is because it's influenced by characteristics in the ecosystem, like habitat complexity or habitat heterogeneity. Here we have the volume of foliage at different levels in the canopy, and we see that the number of pairs increases as you increase the volume of that foliage. Diversity is a little bit different. It's going to encompass richness, but also the representation, the relative abundance of each of those species. So now we go back to community A and B. Yes, each of them have 5 species. That's the richness. But to look at the diversity, we have to work with an index. Now, don't worry, we're not going to go through this. I just wanted to show you the index. There's lots of different versions of this index, but this is what's known as a diversity index. It works exactly like a GPA. There's an identity, which we are, in our case, is species. And then there is an abundance count. And so you'll see that at the end, you end up with a number that allows you to see the GPA of these communities. And of course, like a GPA, this index increases one of 2 ways: more species or better representation of those species. And it also goes down for the same reasons: less species or poor representation, which is the same thing as one species dominating that space. So we care about diversity as well, because we know that it too is influenced by characteristics in the environment, like habitat heterogeneity. And you can see that, again, positive relationship between bird diversity and the diversity of canopy layers, of foliage layers inside that canopy. The more diverse the canopy, the more diverse the community of birds. This one's an interesting one because here we're looking at the species diversity optimization actually occurring at intermediate levels of disturbance. I know that you probably thought that disturbance would be something like linear, but it's not. It's optimized right in the intermediate levels. And that's because too much disturbance means that species can't respond quick enough. So there's a drop in the diversity. Too little disturbance, and then you get a few species that tend to dominate that space and don't allow other species to come in. It's only in the middle where you have a balance between these two forces. So now that we know how diversity is constructed and why it's important, I think biodiversity is the next term that I want to make sure we're all using the same way, because it's actually a construct of 3 elements. One is genetic diversity. So how many genes and how well represented they are. Now remember, with genes, they're not sitting still, they're moving between populations within that population, what we call a metapopulation. And of course, this organizes itself spatially also in terms of, we're interested in species diversity as well when we talk about biodiversity. And then the last one that we want to be focusing on is diversity of ecosystem functions. This one's a little bit different. I'm not sure if everyone is as familiar with this one, but these functions play key roles, especially if they're not redundant in an ecosystem. If the one species is responsible for doing it, it's a big deal when they're gone. So I wanted to highlight an example of an ecosystem function. And I picked an example from the Pacific Northwest. These are salmon that are coming in from the ocean to spawn. And then of course, when they finish the spawn, they die. And it turns out that the nutrients from the fish dying off, they're not all consumed. They stay in the rivers. Those nutrients are actually uptaken by the trees around those riparian areas. So here you have 2 data sets. One is where spawning salmon are allowed to enter the river. And then here are rivers where the dam has not allowed the spawn to occur. So it's blocked. And the other part that I want you to focus on here is this 25-meter range within the edge of the river. And what you can see is that when the fish are present, when the salmon are present, These are the trees that get the biggest basal area, the biggest growth. But when the spawning is not allowed to occur because of dams, notice that those trees near the edge don't grow as big. They get smaller basal areas. What's also interesting is that as you move away from the edge of the stream, this riparian area, it doesn't matter whether the fish are present or absent. It's exactly the same growth. The nutrients don't make it out that far. So in this particular case, the ecosystem function, this salmon swimming in with marine nutrients, making them available to the first 25 meters of the riparian zone, is nutrient import. It's a nutrient importation system, right? That's what the function is. Now, sometimes this feature, this function in the ecosystem has a special bonus, and that is that it benefits human health. When that happens, it gets a different name. So when you have an ecosystem function that benefits human health, we call it an ecosystem service. This is the holy grail. Everybody wants to be able to identify an ecosystem service. Why? Because when you do, you're benefiting human health at the same time. So it's a great widget to be able to identify, which again is an element of ecosystem function, which is an element of biodiversity. So here is an example of an ecosystem function that benefits human health, and that is the more street trees in this particular neighborhood, because it really depends on the species, actually allows asthma prevalence to decrease in children. So here's a really cool ecosystem function, which is capture of particulate matter. And in this case, it just happens to benefit humans, especially children, that now don't have this prevalence, this constant asthma symptoms. So let's shift now to some of the examples. And remember, when I'm going to show you these examples, we're doing this integrative approach. Remember, there's two steps. Step number one, we're interested in studying the group of organisms or the species and that population. And step number two, we're interested in analyzing its role in the functioning of that ecosystem. So there's a bit of a dive into once you identify the group. So let's take a look at this data set. This data set was actually collected by students in Trio Upward Bound. This is a summer program. Last summer, it was fully virtual. So the students that worked with me got to do some GIS and some data mining, all virtual. So here's some of the data that we got to dive into. We were interested in green. We wanted to see how green at county level changed with respect to asthma. So very much like the example that I gave you for ecosystem services, we wanted to know if we had these ecosystem services present here. So here is on the white axis, forested land as a percent of the county area. And down here, we have persistent asthma. This is everyone, right? Everyone with persistent asthma, not just children, it's all adults as well. And what we see is that there's this negative relationship. The more forested your county is in Michigan, the less persistent asthma as a part of the population we see. So very much in line with what we just saw as an example. Forested land as a percent cover matches this well. Then we decided to make a quick change. What if instead of looking at forested land, we grouped it in a different way? There's other ways to organize forest. And we decided to go with the state parks. So in this particular case, we're looking at the size of the state park in the county as a percentage of the county. And we found a disturbing relationship with the number of emergency room visits by children with asthma. There's actually a positive relationship here. In other words, the bigger the state parks in your county, the higher the number of children with asthma making emergency room visits one or more times. That's the relationship we detected. Very different from the forested land. We then changed to the number of state parks, not the size. Now we're just counting how many state parks there are in your county. And again, we see this positive relationship with the number of emergency room visits by children with asthma in your county. So there's this association between these two variables. So this puzzled us. Why does forested cover give you this ecosystem service identity? But when we look at packaging green as the state park, differ. And this kind of made us think a little bit about how state parks are designed. And so this launches the next layer. And again, this is the nice thing about integrative ecology is we found the phenomena and we're describing it in terms of its ecosystem function. Now the question is why? So this is where the engineers would come in, or in this particular case, landscape architects. Maybe as a start, it would be great to explore if landscape architecture, how these parks are designed, could be or is, or what that role looks like in playing, in contributing to this association that we see with children with asthma. But that's the direction that this integrative approach would point us to, now that we've identified it. So I want to use this again as kind of a stand-in, right? Step number one, what's the group that we're studying? And step number two, what is the role that that group is playing in the function of that ecosystem? And can we use it, right? How does it work? So in this particular case, we were interested in biogeography. We were actually, this is conducted in El Salvador. And we were interested in using a tool that measures landscape elements. In this particular case, we're looking at something known as patch shape. So this formula gives you a value whenever the shape is close to a circle. So for example, these would be landscape elements that have an S1 value. They're close to circular. But as you move to the right here, you can see that those now have more perimeter to area relationships. So we'd expect that value to be larger. It's a neat trick, very easy to measure, but it also lets us play a little bit with some of these landscape elements in a biological sense. So we decided to work with urban green spaces because we knew that in San Salvador, which is huge, there is heterogeneity in shapes and sizes. So you can see that some of these parks are pretty much corridors across riparian areas, and some of them are quite large. And so we went through and we picked different sizes, and we were, again, interested in island biogeography as island habitats. And we got a nice little paper out of this. What we found that was really intriguing was one particular element in the landscape, which was shape. Remember, here is the perfect circle. Here, as you move away in this axis, you're getting more oblong. So this number probably looks more something like that, whereas something more circular would look something like this. So what we're seeing is that the richness of birds that are in that park, irregardless of the size of the park, just the shape, there was a negative relationship with the shape of the park. The more circular the park was, the more species of birds that were frugivores. So why is frugivores important? These are the birds that are bringing in the seeds. Remember, they're defecating those seeds that it will eventually replace the trees that are in the canopy. So this is what we call in ecology seed rain. So we're actually seeing that seed rain would be impacted by the availability of species of birds that disperse fruit seeds. So what's interesting here is that folks that design green infrastructure and cityscapes they don't usually design the park. It's whatever land is left over. Here, what we're finding is that the shape of that design plays a big role in which species arrive. What we think is happening, if I can backtrack here, is that in a circular spot, the edge effect where there's high predation is optimized away from itself, right? It just effectively reduces the area of the patch. But if you have something that is oblong like this one, that edge effect almost nullifies any effective area away from those predators. So we think that there's a really interesting edge effect here. But in terms of, okay, we've identified the group and what function they play in the ecosystem, this now brings us to the question is, look, can we optimize park shapes in cityscapes so that we can actually sustain the constant seed rain that will replace the trees in that canopy. If you can, then there's a lot of effort right now being invested in making seedlings, growing them out, then transporting them, getting volunteers to then coordinate school groups to then volunteer and go out. Those groups now could be monitoring bird species instead to identify other ecosystem functions. So we could repurpose all that energy if we were able to just incorporate into the design some of these landscape elements that benefit seed rain. The next project I want to highlight is one where we worked in a planted plant. It's a tree plantation. This is actually in Costa Rica in the Atlantic rainforest. The reason I'm highlighting this is because this is not a natural forest. This is a planted forest. When this picture was taken, it was about 15 years old. So I'm thinking bioswales, where you're planting trees and you come back at that bioswale 15 years later. The idea here is to use a tool where you track canopy temperatures. We climbed up into the canopy and placed data loggers to track the temperature and compare it to a forest that was actually regenerated naturally. And so what we did was we compared two forest types. One was planted, one was natural. And within the planted, we had two different subsets. One was one species, a monoculture, and the other one was a polyculture, 5 species planted together. The monoculture was actually planted with the dominant species in this ecosystem. It's a legume. If any of you are familiar with Costa Rica, this is the pentaclathera, this beautiful tree that's common in the lowland rainforest. So the graph that I'm showing you here, there's only a couple of places when I know it's very busy. I want to show you a couple of places to look, is the hours. The ones that are with asterisks are the places where we saw differences between the secondary forest, which is our comparison here, and the other two planted forests. But what's interesting here is that the monoculture, which is the green, monoculture, which is the red, this group right here corresponds to something we weren't expecting. The monoculture only differed from the secondary forest, the natural forest, in 1 hour of the day. That was the only time. The rest of the day looked exactly like a secondary forest. The polyculture actually differed for the remainder of these hours. So 6-8 hours difference. We expected the polyculture to behave more like a natural forest, not a monoculture. And then we remembered, that monoculture was planted with the dominant species in that forest. So fast forwarding now to bioswales. What if we were able to use the optimization of tree canopy temperatures for the species that move through those? I'm thinking arboreal mammals, so that they look like a natural forest. Well, we would have to know, are there dominant species that play a big role in shaping that biology? This would be a great way to make bioswales feel, at least temperature canopy wise, more like a natural system. And the last one for the forest that I want to highlight is with gene flow. And that's because we did some work. This is actually here in Detroit, in Palmer Park. We were interested in studying cohort structure. This is a survivorship study, classic, static life table. And we wanted to use this census data, how many trees and how big, to monitor the stability of the trees in this forest. Because remember, if you're familiar with Witherell Woods, it predates the park. The park was established in 1893. This forest basically was there before the urbanization occurred around it. So it's a bit of a fragment that's been captured by the park. What we found was that the case-selected species, these climax species, these late successional species that are in the park are not replacing themselves. In other words, if you can squint here, for red oaks, there's nothing smaller than 20 centimeters diameter. For black oaks, there's nothing smaller than 25 centimeters diameter at all. No seedlings, no trees smaller in diameter. We did find a few between 5 and 11 for beech, but as you can see, there's no way that that's going to replace that crowd. Think of it as high school. If you don't have a lot of seniors, you have very few seniors, and then you have a spike of sophomores, and then you have no freshmen coming in. That's not a stable population, right? That means that when the canopy trees go, there's nothing growing underneath it to replace it. So because this set of trees predates the 1800s, right? 1890s, I should say, this park represents a snapshot of genetic diversity that is no longer able to leave the park, right? Acorns are not going to cross woodward. So we think that the best way to approach the sustainability of that park isn't just planting trees. We want to recover that diversity. So we actually think that maybe anthropogenic dispersal of those trees using cloning or grafting tissue samples, you can actually grow up clones and now go and plant them in other parks so that you can recover some of that genetic diversity that is going to get lost once those trees go. Shifting gears now. Oh, sorry. Yes, right. This is the last thing I was going to mention is, remember, this is not going to be a really quick solution. Planting trees anthropogenically has one problem. You have to plant a lot. Trees tend to follow what are known as type 3 survivorship curves. Plants make a lot of seedlings because, as you can see, there's a high mortality of seedlings early on during the early cohorts, and only a few make it into adulthood. So if you are going to do something like tissue culture, it's going to be a lot because not all of those will mature into big trees. So it's a tricky compromise. Switching gears to urban water, this part is a little more fun because here we're now back in El Salvador. One of the big problems in urban rivers is they're always polluted. They always have this contamination of solids as well as chemical effluents. And sometimes there's an overzealous approach in dealing with the abiotic and biotic problems, dredging, removing, etc. And sometimes in that approach, we forget interactions. So I wanted to show you in this particular riparian system in El Salvador, we actually worked with different disciplines that were treating all these polluted rivers. And we jumped in and decided to take a look at the biological element of the story. And so this was our little contribution. We got a nice little paper out of that too. We took a native molly. It's what you find in the pet stores that grows wild in the streams. You can see that these streams have garbage. And in the back here, there's actually gray water coming out of a random house in the back there. The smell was not the best, but these fish are thriving there. They're actually incredibly robust. We grabbed them and we placed them inside Dixie cups, filled them with water. We fed them the first day, and then we left them for an entire 30 days without feeding them. When we came back, this is what we found. Where fish were present, the mollies were present in the cups, there were no mosquitoes. Wherever the fish were absent, because we didn't put any in our controls, you could see we got lots and lots of mosquitoes, larvae swimming around. The cool thing is we got a little video of the fish. We threw some mosquitoes to make sure that they actually could eat them and that the mosquito was not afraid of laying eggs in there. And you can see these mollies just go to town on these mosquito larvae. And so this was a big surprise because in El Salvador, there is only one university with a biology department. There's no biology department in the entire country except at the state school. And so when folks work with daylighting rivers or urban greening, they don't have any scientists. And so the idea that you couldn't just sterilize water to clean it up, that there's actually organisms that could keep in check pest species was a big surprise. But this made me think a little bit about a similar habitat that we have here in our backyard in Palmer Park, where we have salamanders that are common to Michigan. These are common and they thrive in Palmer Park. We know that they eat mosquito larvae. The question is, can we quantify it to see if it actually is playing an ecosystem service, right? Because those mosquitoes are no longer transmitting viruses. In El Salvador, it would be dengue, Zika, chikungunya, Zika, and of course here it would be West Nile. But this would be something that could be quantifiable, this idea that an organism could be playing this type of ecosystem role in our backyard. Keeping with that theme, with wetlands, this paper just came out. I don't know if you saw it on the news. It looks like lizards could be keeping in check Lyme disease by keeping tick populations in check. And what I wanted to show was a California study that we ran. This is my home state in Southern California, where we worked with the California fence lizard that's common in the coastal sage scrub, which is the low elevation chaparral, the soft chaparral as they call it. You notice that the California fence lizard has this beautiful blue coloration, right? That's how they mark territory. That's how they attract mates. And that's how they kind of show that they have a good body condition, right? So we captured a few and we took pictures of the underbelly and we measured the pixel intensities of these underbellies. And then we snipped tails from these and we ran them in an inductively coupled plasma spectrometer to look at heavy metal concentrations, because this beautiful landscape you see back here used to be full of oil rigs back in the 1800s. This is where they would extract, well, Getty used to extract petroleum, and that's where he made his fortune. So that's what that marsh used to be. And when we ran the numbers, we found a relationship between different metal concentrations, they're all listed here, and the color intensity of those patches that the lizards had. So remember, these patches are how the lizards attract mates, sexual selection. Heavy metal concentrations in the animals is correlated with changes in the coloration, making them more drab, less attractive. So what that means is areas that are contaminated with heavy metals aren't just toxic for wildlife, period, right? But those animals that happen to be eating insects that also live there are collecting those metals, and they are now less likely to theoretically here is they would have a fitness outcome, right? They were not going to be able to reproduce by not being able to attract mates. If their coloration is off, it could be read as a body condition issue. So this idea of bioswales, because what you're looking at here is a bioswale. This is basically runoff, stormwater runoff from the townhouses that are right across the street. You could actually see that contamination could be a series of spots, contaminations that could be affecting different populations. So the key here is we don't know if interactions at these different spots are being affected. And the key is, can some of these spots serve as sinks and sources for lizard populations that are not reproducing very well so that they can replace themselves? So we can now think about these bioswales as a network of sinks and sources for organismal populations to move around that can provide ecosystem services. For example, the study with the ticks. The next one that I want to show you is with anthropogenic irrigation. This is more of a drought tolerant ecosystem, but the idea is the same. Take an ecosystem that's adapted to having long winters like Michigan, and winters now start to shrink. How does that habitat now react to that? So I picked it just for this reason, because in this particular neighborhood, they had a sprinkler system installed in the neighbor's xeric landscape. So they had this beautiful coastal sage scrub, but they installed a sprinkler system. So we used it to take a look at how irrigation affected the flying insects that would visit flowers, so we were tracking pollinators. And what you can see here is two different communities, one that's irrigated, one that is not. And the pie chart, if you look at the patterns, represent the same species. So the same color pattern is the same species. You'll see that not only do they change in size, but they also change in representation. So irrigation has incredible changes, produces incredible changes in the insect community. And these are flying insects visiting flowers. So potentially most of these are going to be pollinators. So what this brings to mind is, think of bioswales as successional stages, right? If you're installing a bioswale, can you pick species that are adapted for that? In other words, don't plant trees that don't have the biomass in the understory, the organic layer in the soil is not yet there, the nutrients aren't there. Let's start it as a successional stage. Can we stage those in our planning of how we engineer bioswales? The last one for this section is one that I talked about in my lightning talk with Healthy Urban Waters. And it looks at fecal indicator bacteria and how they respond to 2 invasive species out in Metro Park, Lake St. Clair. And the Metro Park has been rerouted to empty, not at the beach, but rather through a series of bioswales and then to a marsh. And all of these have these invasive species. The first thing we wanted to do was, let's see how bioswales behave with fecal indicator bacteria in off-season, right? This is during the fall. This represents right here the EPA limits. As you can see, the coliforms and E. coli all were at or above the EPA limits. So they were above and exceeding, meaning that these bioswales aren't really containing the FIB communities. But what was really surprising was the 2 invasive species in those bioswales are effectively reducing or stabilizing the E. coli communities. We don't know why. We just know that those invasive plants are stable. Whenever we saw the plants away, immediately the levels shot up and it just became sporadic. So we think that there's an ecosystem function that's being recovered. We don't know what it is yet. We wish to identify it because technically you could swap it out for a native or try to find something that's not invasive that actually could sustain that inside your bioswales and stabilize E. coli communities. The last section, which is the short one, is with urban agriculture. We've been tinkering with these aquaponic systems to try to mimic what bioswales do. I'm thinking open system and closed system bioswales at different scales. Rather than work with the big ones, we work with the smaller ones. And I just wanted to highlight 2 projects. We grow plants in these aquaponic tanks. And this is actually a system that was co-developed with Adrian Vasquez. And these 2 student groups found something really intriguing that's adaptable for green infrastructure. One group with acid rain, because we know that in Detroit, we actually have the more acidic acid rain for the country. And they also worked with road salts because we use road salts, effectively magnesium road salts. And so the 2 students set up these different solutions, aquaponic solutions, to expose the onions, which grow really quickly. And we compared root performance in these 2 systems. And I want to share quickly the 2 data sets that we found. Most of the acid rain was sulfuric or nitric acid or combinations therein. We even had point sources. But what was surprising was this. Here's the control, no acid rain. Here is a low concentration sulfuric acid solution matched to the acid rain you'd see in Detroit. And what you can see is that the roots actually grew longer in the treatment. Then if we look at the salt treatments, there was one in particular, magnesium. It's a positive relationship. All the other salts had a negative relationship. We actually see an expansion of root length with magnesium salt as well. So here are 2 elements that are basically in the literature showing that they're able to expand vacuole inside of root tissues, and it's causing the plants to chemically induce growth. So the consequences here are, if you're a plant and this is supposed to be your budget for growth, but now you have to, this was your budget for growth, but now you have to expand it, it's going to have to come at the cost of something else. So we're now working with an urban garden in Southwest Detroit because we think that what is going to be compromised is plant defense. So we think that urban gardens being exposed to these acid rains and potentially road salts like magnesium would be more susceptible to lower defense plants, meaning they would probably experience more herbivore or leaf chewing insect damage and likely have to use pesticides. as opposed to their rural counterparts where they don't have to deal with this. This last project is just for the Q&A so that you guys, FYI, this is one I'm still working on right now. We're collecting the pollen baskets from honeybees because we're trying to identify pollinator deserts, areas where honeybees are not collecting enough resources to sustain populations. And we're trying to find out if we can direct, rather than invest in greening where there is already greening, let's direct greening to these neighborhoods where these bees can't do their thing. So that means that you can't have apiaries for honey and you can't have home gardens that require pollination. So my last thought as we kind of wrap up here is obviously collaboration is a really big part of what I do. And I wanted to pick an example of how I feel collaboration really works for me because it's a little different. This is an exercise that I do with my freshman classes. It's an introduction to evolutionary biology. I give them 2 rubber bands and 2 straws. And I say, okay, make a freestanding structure. And they all sit down and try to make something stand up. Then when they finish, I give them another straw and another rubber band. And one group, I say, hey, can you guys add? Don't take apart your structure, just add to it. And then I tell the second group, okay, you guys take it all apart and rebuild. Now make it stand with all those rubber bands and all those straws. And an interesting thing happens when I do this. The group that rebuilds has very, very little variation. They all converge on let's make it stand. But because the group that got to just tack it on to something that was already standing, they got to be creative. And so there's a lot more variation. The reason I'm pointing this out is I work with a lot of engineers and I'm a scientist and I look at this in a very different way. I look at it as if there was selection against being too tall in this environment, look what happens to my rebuild community. They disappear, they go extinct. Look what happens to my add community. They get to stick around because they have variation. In order for us to recover ecosystem processes, we're going to have to add redundancies into these engineered systems. And the only way we can do that is collaboratively. Thank you.
Thank you, Dr. Carmona, for that talk. At this time, we have an applause. We will
Thank you.
We will take questions now. I think if anyone has questions that they have for Dr. Carmona, now is the time.
And I don't think I can see the questions. I think you get to see them.
Okay. All right, so I'm not seeing the questions myself, so I don't know if Delashan or Jamie are the ones seeing those.
So currently there aren't any questions in the chat. We do have some comments from individuals who wanted to thank you for an excellent presentation. Apparently they're an environmental science instructor at 2 community colleges. But if anyone has a question, please feel free to either put it in the chat or just unmute yourself and ask your question of Dr. Carmona.
Yeah, and thank you, everyone. I'm seeing some comments through direct messaging, so thank you for the kind messages.
Okay, let me see if I can look at
and I try to pick
Carol Miller has some questions. You can just open your mic and ask them, Carol.
Yeah, okay, thanks, Adrian. And Victor, thank you so much. That was such a fun, enlightening presentation, even though you sort of took a swipe at engineers at the very
No.
it was still wonderful. I appreciated your your comment about diversity in design or redundancy. I guess we often call that resiliency in design. I thought that was a great example. Just curious about any comments you might have about coming to Detroit to teach in our wonderful backyard. It sounds like you've got great things going on in this region, but I'm wondering how was that for you in terms of a transition coming from Southern California and some of the other more exotic places, I guess I'd say, or more tropical sort of places?
Yes. So thank you. And I didn't mean it to come off as a swipe at engineer. I work with so many. I feel like
it's all good.
I have to speak up, because I'm in the corner like, wait, redundancies, which is a bad word, right? I wanted to talk about first about the redundancy because I actually got a question about that. So redundancy means an ecological process that's redundant. So there's several species doing nutrient cycle or several species doing that particular process. They're redundant in that system. And ecological redundancies are important because if one species now leaves, the range changes or something happens, you still have them present. So that's what redundancies allow you to do. In terms of resilience, the way we look at resilience ecologically is in your ability to bounce back from change. Because remember, change is constant. So here it's seasonal, right? So you've got to be able to come back. If you can't get back from a disturbance, then that's not being resilient, right? That's usually a bad thing. So I wanted to touch on that first, on what resilience was, because that was one of the questions that came up. And then in terms of the biggest changes, the thing that I really liked about Detroit was how voracious the city is in green infrastructure. That stood out to me because in California, I got to work on a couple of FIB projects, especially with respect to antibiotic resistance. And those were really exciting because I got to work with all sorts of folks. Microbiologists and ecologists don't always, they're not even on the same floor sometimes in their own buildings. So, and plus all these engineers. And I just found it absolutely fascinating that you could look at a bioswale as an ecological feature, like a riparian area has leaching, that you could actually do that in a bioswale. So that was my first introduction to it, but there weren't that many. Usually they were quite expensive to make. So only a few places made them and they only had one, n equals 1 is not much we can do to study variation. When I came out here, there's a lot of them. I mean, with Adrian, we just ran that study that you helped us out with. That was like being at a candy shop. It was just, there's an incredible amount of things that can be explored there. And what I also enjoy about Detroit is in that same space will be other disciplines working as well. And so it's very easy to kind of gravitate towards some type of collaboration. And that's how I, in fact, met you, was in that exact same atmosphere. So I have enjoyed it very much in terms of the transition. What I've done is, as a good biologist, is when I'm in Costa Rica, I usually work on one type of biology. When I'm in El Salvador, I work on another. When I'm in Michigan, I do more of this type of green infrastructure. But when I'm in California, I work on other systems. So I kind of adapt to where I'm at and where the funding goes, just because there's so many interesting questions at these different locations.
Thanks so much.
Thank you. And let's see, I've gotten a couple of others, Adrian, other questions.
Yeah.
Let me see if I can scroll through it. Oh, right. So I'm going to go back up. Barb is asking me about redundancy, how it's achieved. Well, in natural systems, it's present already. There's several species that do similar processes, right? We call them ecological niches. It always isn't just one species. There's several ones occupying that particular niche. So they may carry out similar functions like detritivores or frugivores or, you know, or nitrifying bacteria. We classify them by their function. And so there's several organisms that will, or sorry, species that will carry that out. So those are redundancies. The key is, can you identify them? And in these anthropogenic systems, like for example, bioswales and green infrastructure, I think it's important for us to kind of make an inventory, right? It's like, how can you run a shop if you don't know what you have in inventory? So identifying those redundancies or lack thereof might be important, because you might be bringing in some ecosystem function that is important, right? You don't want anaerobic bacteria growing in your bioswale. So if you can reduce those would be great, and have them be redundant. That makes them more resilient, as Carol Miller was pointing out. Oh, okay, let's see. There's another question here. Oh, okay, yes. This is for Teresa. Sorry, Teresa, I can't see your last name. Oh, Ramsey. Yes, Teresa Ramsey. Absolutely. Feel free to send me a message. I believe my information is on the Wayne State website. If not, I can also add my email here at the chat and send it to you. But yes, absolutely. I have no problem following up and checking in with you on specifics. Yep. And let's see, there's a question from Lisa Kraus. Yes, exactly. I agree. That's exactly redundancy is. She writes, I think of redundancy as a backup, having a backup plan to achieve the same ecological need. That's exactly right. That's a textbook definition. Thank you. Thank you, Rob. And those are the questions that I have on my end. I don't know, Adrian, if you have others.
Yeah. There was a question from David Pitts. Has the influence of habitat shape and predation been examined with insect populations? Would the circular habitats still have similar effects?
So we did do a parallel study with lichen. I had a group of students that were, lichen are really tough to identify because it's actually two organisms growing together. And so this particular group was really good at going through that key. And so we did lichen in the same parks that we did with the bird data. And we compared two substrates. These were lichen growing on tree trunks, on the bole of a tree. And so we had exotic trees. Mangoes are actually from India. And so they're growing in Central America. They're exotic species. They're not invasive, but they're exotic. And so we wanted to compare an exotic substrate versus a native substrate. And we went with one of the local species, and we did it across the same network. Again, our objective was to kind of look at island biogeography inside of this matrix of green spaces in this urban space. It's, you know, San Salvador is just massive. It's kind of like Sao Paulo. It's huge. It just goes on and on. And so we were very surprised that with that particular organism, a mutualism, there was no response to the size of the park or the shape of the park. So just with those three taxa, I can tell you that it's taxon specific. So the thing about lichen is that they're important for fixing nitrogen, right? We saw the biggest difference in the lichen communities with respect to native and exotic substrates, but not with the shape of the park or the landscape of the parks, you know, how big they were and how far away they were from each other. So the way to answer your question ecologically is this: lichen, we don't see a response at the landscape level. Birds, we do. With lichen, we see them at the substrate level, meaning the community or even population level. So these are two ecological scales at which each of these species are responding. So different species will respond at different scales. the job of the ecologist is to identify the relevant scale. So I hope that answers your question.
Hi, this is David.
Did you answer Gloria Rivera's question?
Yeah, that's very interesting. I was also thinking about, I have a graduate student who's going to evaluate pollinators at green infrastructure sites in Detroit area. I was thinking about shape as another variable to pay attention to.
Right. So are you sticking just with pollinators, period?
I think that's still under discussion. This is stretches my expertise definitely, because I'm a pharmacologist. So.
Oh, I was gonna say I've done insect work, and in fact, this summer I'm doing some not similar, but the idea of insect diversity in urban landscapes is equally fascinating to me. So I would love to help you solve this problem. I think it's worth collecting because remember, I did this in a tropical system where birds are feeding on fruit. Clearly, that guild is not a big, it's not represented very big here. Right? There's going to be seed-eating birds rather, or insect-feeding birds. So I'm thinking the same thing with insects. You're going to get different taxons behaving in temperate biology one way and very different from the tropical. So something that I wanted to kind of pitch as I was going through these examples is they're all highly tuned locally, right? This is not one size fits all. So it's kind of like the rubber band example. If the architects and engineers make the structure, the couple of rubber bands, and then the scientists come in with a couple of more rubber bands and add it on top, we can create a lot more craziness. Does that make sense? So yeah. I think that your plan is a good one. I say go for it. And my advice is use Google Earth. That's what we ended up doing. We used Google Earth, and then we measured the perimeters with some image analysis software. It was really nice. It was really accessible.
Yeah, I'd love to talk to you further about it.
Sure. Feel free to drop me a line.
Okay. Thank you.
All right. Did you answer Gloria Rivera's question? I think she had a question. I don't know where she sent it to.
Who? Sorry, Adrian.
Gloria Rivera.
Oh, Gloria. Sister Gloria's on here.
She had a question, but I don't know what the question was. So maybe she sent it to you.
I haven't seen it on my... I'm getting direct messages too, but I don't see the general messages.
Okay, well, if Gloria could ask it again.
Or do you want to unmute?
I am unmuting.
Hello.
I put it in the chat, and maybe it was kind of wordy, but at the end, the thing is, we have two disciplines, engineers and environmentalists or scientists. And my question is, do you have any examples, and maybe you named them and I missed them, Victor, of something that's going on that is beginning to find a point of opening up to each other and including, kind of like the example of the straw. If I am, is there something that that's happening here in Detroit, or beginning, or even in the thinking stage, where the two are beginning to realize the value of this collaboration and the incredible richness of it? That's essentially my question. I don't know if it's clear enough.
So it's typically, and thank you for mentioning the rubber band example. I wanted that example to play that role. The idea of, you have one discipline come in with a set of tools, and then you have another discipline with another set of tools, and you create something bigger than life together, right? Rather than reinventing the wheel within each discipline. That's exactly what I was going for with the rubber bands. And the first example that comes to mind is one that I just ran with Adrian Vasquez and Kara Miller. And that was with my class. We went to the Metro Park. And we used, it's kind of like a, well, you know me, it's like a mesh of all these things. It's a class that's learning to do research. And so they set up these independent projects, but I made sure that they went to several different bioswales. So they set them up. I have copies of all that data, and I reanalyzed it to look at how those invasive species altered the levels whenever they were in those bioswales. So, it was a design within a design, but those bioswales were constructed for a whole different purpose. Their purpose was to take all that stormwater runoff full of the bird feces from the parking lot and divert it away from the beach, which had to close all the time. And so they redirected it to go through these, it's beautiful landscaping features all the way down to the marsh. And so it's, it's a wonderful park. Like I was saying, it's a beautification kind of, you can go to the beach now and you can now walk your dog through these, and it's brilliant. There's, birds. It's a lot of cool wildlife there. I've even seen deer. But beyond the beautification, there was this open invitation from Healthy Orbit Waters, which was, hey, do you want to do something technical within this bioswale? Go. And they supported me like no one has. It's incredible. And so we went there with the class. The class was blown away. They've never been at a research facility for graduate students before. That was all new for them. And for me, it was fantastic because I've been able to share that information. And we found now some preliminary data. You met her. One of my students, Cynthia, is applying for funding to go back this summer during the peak of the activity of bacteria where it's warmest and rerun those sites. We have them all mapped so she could go rerun them and see how it alters. But the nice thing is it's all pointing to these invasives doing something unexpected, right? The invasives, you want to remove them. Just the word, it already tells you you don't want them there, right? They're aggressively expanding their range. They're not leaving enough water for the waterfowl to come in, and they're being removed actively. But the fact that we identified some strange function that seems to be making more consistent E. coli, then two questions that come in: what is that function, and what exactly does a more stable E. coli community mean than an easier one to get rid of? I'm thinking like picking up a mess versus a small contained mess. It might be easier. So there's more ecological explorations to do, but clearly I think that if we're able to identify that feature, it might not be, I'm thinking like a list of plants that contribute to ecological functions in bioswales in Detroit, Michigan. Like you start making a list. Here are all these plants that do this stuff. And now when folks go to plant, rather than saying, let's just plant natives, now they can say, well, let's plant natives that are selected, these early successional species. Let's plant these because we need to make sure that we have redundancies and these types of, look, there's lots of birds here. How do we do this? So I feel like there's a cool, good, the hard part has already been done. Now it's a question of let's identify it. And that's the fun part for the science folks, right? But I think there's now a hunger from the engineers of hurry up and tell me what that process is because we want to use it, right? So, and then the one that's being explored is Palmer Park. Palmer Park is going through a revitalization right now, and they're talking about making major changes to Witherell Woods. And I'm a little worried about the genetic diversity of those trees being lost, the salamander population being restricted. It's a very narrow habitat range as a proper herp would have, something very, very small. And they're doing it to beautify it, which I think is fair, right? Who doesn't want a beautiful park in their neighborhood? But at the same time, I'm trying to, I go to the committee meetings now to try to bring up that, hey, let's not forget the ecological systems that need to be recovered within those green infrastructure projects that are being funded there. So there's an ear for this, for sure. I'm seeing an ear for this type of conversation. I haven't been turned away, which is great, which is really, really great.
Well, thanks a lot, Victor. We're out of time for more questions, but you can contact Victor at his email. I think it's what's your email again, Victor?
Oh yeah, I'm gonna put it on the chat to everyone.
Yeah, to follow up with you. So we're at the end of the event. Thank you again, Victor. Thank you for that talk and all the different projects that you're doing in Detroit as well. And I just wanted to tell our audience that the next WIN at Water at WIN seminar will be on April 1st. Dr. Krista Wigginton will be discussing how environmental engineers have helped address COVID-19. And the recording of this event will be sent out after we're finished. So thanks again. Thanks
Thank
to
you.
all the participants.
Thank you.